Thank you. All right. First thing first, can you hear me? Okay. I have a pretty loud voice. Is this microphone doing anything? Okay. I can't tell that from here. Anyway, thank you, Dan, for that introduction. Thanks, Dan and Ben, for the invitation. It's been about 10 years since I was up here, but it's been great to reconnect with some old friends and meet some new folks as well. This is a fantastic place, doing a lot of amazing things, and it's got, of course, a very rich history in ornithology, my favorite topic. So what I'm going to be talking to you about today is essentially an overview of some of the research projects that we have going on in the lab. And as Dan mentioned, yeah, it's pretty bird focused. I think I might have been on one mammal paper one time, but it's almost exclusively birds. And the range of topics in my lab sort of span higher level bird phylogeny. For example, we have a new grant to basically reconstruct a phylogeny of all bird species. So if any of you out there are interested in analyzing data sets that are extremely large on birds, you let me know, because there's like a zillion spin-off projects on that to do. But I would say my true love is really down at the fine scale level, population genetics and hybrid zones in particular. So I did my PhD on hybrid zone in western Panama between two species of manakins. These are birds that have secondary sexual characteristics, and the plumage characteristics of one of the species had introgressed unidirectionally into the other species, presumably due to some sort of selective advantage. So another theme in my research is I'm really very interested in trying to connect the natural history of birds, sort of what they do on a day-to-day basis, how they eat, how they move. I want to know how that affects their speciation, their population genetic structuring, and so on. All right, so this is a barred ant shrike. I took this picture in the Yucatan Peninsula on a little family vacation with the kids last year. It's a bird that is widespread in the Neotropics, okay? Pretty much every place in South America you can see this thing, and it gets up through the Yucatan Peninsula, it gets to Chiapas, and then it stops, okay? And this is a bird that can live in super hot, pretty cold. Why isn't it here in the United States? And that's one of many questions I will not be addressing today. But I do love to muse over distributions of organisms. All right, so I think it's good to try to put your research in a really big context. And so I think this is one avenue where a lot of the research that we're doing in the lab fits, although most of our work is in tropical regions. Why are the tropical regions of the world more species rich than temperate regions? This is one of the big questions. It's got a very complicated answer. Early on in my undergrad days, when I was just trying to determine whether I wanted to pursue grad school, I was a field assistant with a PhD student down in Peru. I took off the fall semester, and basically, his PhD was getting at this last part, the fact that there are tropical species that have really no ecological counterpart up here. And the birds that we were studying were these army ant following ant birds. And I assure you, none of you have seen these up here in Michigan or anywhere around here close. So it's one of the coolest neotropical phenomena. It's a group of ant birds that are completely specialized on following essentially a single species of army ant for food. And they're not eating the ants, at least not intentionally, but they're eating the prey that is flushed by these marauding swarms of army ants. So you got, little katydid, flies away, thinks it's free, the bird's there to chomp it. And it's like a really reliable way to find food. And so if you look at the army ant following ant birds, they essentially come in sort of three levels of specialization. You've got what we call the occasional followers that, they have their territory. The bird has its territory. An army ant swarm comes through. They're not going to ignore it, okay? They're going to, take advantage of it. you've got your regular followers. And just notice, I mean, they're some of the most spectacularly beautiful birds, and a lot of them have bare patches of skin that are pigmented. We don't know why. This is something Rick Prum at Yale has been studying, but no one really has an answer to it. Anyway, they'll follow a swarm beyond their territory. And then you've got what are the obligate followers. These are ones that, basically, if army ant, S. tumbercelli, goes extinct, these go extinct as well. They're 100% obligate on army ant swarms. And so I remember on the ride back from Peru with this grad student, he was like, God, wouldn't it be cool if we had a phylogeny of ant birds? We could like actually figure out how many times army ant following evolved in the ant birds, and maybe put a date on it, determine whether it's like a reversible thing or all these sorts of things. And so I sort of filed that away. I didn't even know what a phylogeny was at that point. And so I'll come back to this later in the talk. All right, so a lot of what I'll talk to you today about is just sort of is speciation of birds. And I always just provide a basic definition. Most of you know what this is: speciation, formation of species. And in birds, although you'll hear about some other mechanisms, I can assure you that most species of birds are formed by geographic isolation of populations, allopatric speciation. You have a population that's continuous. Let's say a mountain arises. These things are isolated long enough, and they begin to differentiate through the drift, natural selection, whatever, and eventually they're going to be different species, reproductively isolated. All right, so this is what I refer to as the speciation cycle, and it's essentially you have speciation, okay, formation of two species. Eventually they expand their distributions to the point that they are ecologically and reproductively isolated and can be completely sympatric once again. Okay, and that's like a full revolution of a speciation cycle. And so one thing that we've done, I think it's a pretty cool thing that you can do with phylogenies if you have good species sampling. So we had a full species level phylogeny of the Furnariidae. This is not the drawer of birds that I show my mom when she comes to the museum, because they're all brown and sort of boring looking, but like ecologically they're incredible because they've diversified into every sort of niche. There's your blue jay, there's your warbler, there's your woodpecker, there's your nuthatch, okay? And so you can basically, if you have a full sort of phylogeny of these things and you know their distributions, you can ask how long does it take for a speciation cycle to occur. And basically, I can just tell you, it takes a really, really long time. What that means is that if you go out into a community, and this is true here in Michigan, it's true in Louisiana, it's true down there, that the average species, the species that are occurring together are very divergent from each other. They're not each other's closest relatives. They're not even cousins. I mean, it's been a long time because it takes a long time for ecological and reproductive isolation to accrue to the point where they can occur in syntope. All right, so the landscape where I've spent probably most of my efforts over the last 25 years, something like that, trying to understand speciation is in lowland Neotropical rainforest. And so this is a sort of a typical distribution of a lowland rainforest bird or superspecies in this group. This is a genus of army ant following ant birds, Echidnapithes. And the thing to notice is that you've got a species over there bicolor west of the Andes in the lowlands, okay, so a geographic barrier between them. Then you can see that the Rio Negro is separating Rafigula and Lecaspis, again, a big river, and then you've got the Madeira and Urubamba over there. So just take home is that geographic features of the landscape are important in shaping bird distributions and are very likely important in speciation. And so I remember as an undergrad, Bob Zink was giving a course in historical biogeography, a grad course that I wasn't allowed to take. But I always was captivated because I thought it was the coolest thing, historical biogeography, like reconstruct the past and figure out what species are related to what. Anyway, I have sort of used this basic strategy, which is to look at a bunch of species, collect genetic data, co-distributed species, collect a bunch of genetic data, sort of phylogeographic data, and basically what should emerge is you should begin to see shared phylogeographic patterns amongst them that reflect their common speciational history. So if they were all out there in the landscape and a mountain arose, then there should be a divergence point, at that time. And then a river arose, okay, and there should be another divergence point at that time. And that was all awesome until we started collecting molecular data. And so that's like the divergence values that you see across 72 species across the Rio Negro. And, it should be pretty obvious to you that they're like all over the map, okay? There's like, it's not a single event, right? And you can put statistics and methods on this, and I can assure you this is not consistent with a single, river subdividing things. It's the same when you look at their individual phylogenies, okay? Like every single one is different, okay? In some cases, across the river is really divergent. In some cases, it's less divergent. So there's like very, very few commonalities. And we've now collected enough data from other barriers in the Amazon basin that the rule now is that there's nothing in common. Okay, every species has its own individual history. So idiosyncratic histories is now the rule. So the idea of vicarious biogeography is, I think it's a good idea. It's probably just premature because we just don't yet have a good handle on things. This is a paper worth checking out if you've never seen it, and it just drives home sort of the complexity of the situation and why you would actually expect there to be very few shared patterns. So what Hayden did was he did a little forward simulation of a series of populations on an island archipelago, little black dots of the archipelago. And so he tried to make it realistic. I mean, this is something you could code in BASIC. So you've got your little island, you've got a little population, you've got migration, you've got local extinction, so a few other little things you throw in there. And so this is a species of sort of low vigility, and so you expect a little better resolution of the phylogeny. That's hard to tell, but just jump right to this, which is basically the situation that most of us will be dealing with, which is just a total freaking mess. There's no way you will ever resolve that, okay? So just, that's okay. I'm just going to live with that. This was good news to me, actually, because you know what? I love looking at birds, all right? I like watching birds. I like feeding birds. And so this caused a shift in my research program. So instead of like trying to figure out what what mountain did that to that, okay, let's take this completely different approach and ask, how is the natural history of birds influencing their speciation? Okay. Things like the fact that birds can fly, right? We all know that, or at least most of them can. And there's clearly differences in the ability of flight from bird to bird. There's a chimney swift, probably not back yet. They're in Baton Rouge. They'll be here shortly. Tufted titmouse, you have those here. But you can see just from their wing morphology that there's difference, captured in their morphology of flight ability. All right, so I was Doug Robinson who's now at Oregon State University now. So we go back to PhD days, both working in Panama. And so we often mused about the fact that there's always been anecdotal evidence that there's huge variation in the ability of neotropical birds to disperse. I mean, you probably heard these stories, like birds of the dark understory will be reluctant to cross even a light gap, in the forest, versus other birds that are just flying all over the place. Doug actually put this to the test. So his student Randy Moore did I can't remember if it was a thesis or a PhD, but basically, this is all animal care and use approved. Captured birds in mist nets, brought them out to the middle of Gatun Lake in Panama, put them in this little box, and then measured how far they flew back to land, okay, and did that to see, what the dispersal ability of these birds were. Let's see if I can get it to play. All right, this is a chestnut-backed antbird in the box right now. This is one of the dark understory birds, okay? It doesn't want to even come out of the box, okay? So she's feeding on the box. All right, please. Ah, there it comes out. Flies. Okay. So that is a poor disperser, okay? That bird doesn't know what the hell is going on when it gets out in open space like that, right? Okay, so they would catch it with a net, release it back into the forest. No birds were harmed in this. Anyway, so I'm not. Yeah, let's keep going now. So, if you take a hummingbird, which they did, you open the box, the thing flies to the land, no problem. So these are just various distances that they released, and we'll just look at this one right here. So this is Myrmeces exel, the chestnut-backed antbird. And you can see just, I guess they gave it 10 meters out of kitty or something, but versus a hummingbird, which just goes. So point is, there's a huge variance here that in dispersal ability. And now the cool thing is we can capture this. We don't have to do that experiment on every bird we study. We can actually use measurements from specimens like you have here of wing morphology to get at this. So these are the birds that they use in their experiment, and these are our measurements, and you can see there's a pretty good relationship there. So now it's something that we can actually measure on birds as a proxy for dispersal ability, and then we can compare how it lines up to genetic differentiation. So this is a study published in 2009. God, that seems recent. But and we've sort of broken it down. These are across different barriers, so the Andes, the Amazon, the Madeira River. And what you should notice is that, and just keep in mind, canopies birds are good dispersers, understory are poor, is that there's just more structure, okay, in the understory birds. They're just more reluctant to cross barriers, and thus they accrue more genetic differentiation between them. All right, we then expanded that study, and this just shows you like the power of like general collecting expeditions. I mean, there's no way in hell you could ever go out and like go, I'm going to do this for my PhD. That would be impossible. But because of 40 years of collecting, we had all of these samples. So, as Dan mentioned, LSU has been, since 1979, been lugging nitrogen tanks to various parts of the world. And we're still doing that today. This is Glaucia Del Rio, one of my PhD students who's working in Brazil. But at any rate, so we had all of this genetic information on 27 species across a bunch of different barriers. And there were two basic patterns that came out of it. The first is that the older a lineage is, and just picture some species that you know, okay? What you find is like the older that thing is, the longer it's been in the landscape, the more sort of genetic differentiation it'll have within it, okay? It basically just means if you want to have more sort of subspecies or variation, don't go extinct, okay? And then the other was just sort of the same result as before, is that the birds that are better at dispersing across will show less structure, more homogeneity. You know, if you're an ecologist, you're thinking, yeah, this is obvious, okay? But hey, we demonstrated it in birds. All right, so I want to shift gears. This is a similar type project, but instead of looking at birds that differ in their dispersal ability or their wing morphology, in this design, we're studying birds that are basically identical in wing morphology. As far as you can tell by their morphology, they would be able to fly identical. But what we did here is compare instead birds that live in floodplain forests, called Varzea, in the Neotropical region, versus birds that are in upland forests. Okay, so a bird like this Plumbeus antbird that lives in floodplain. It basically has the exact same distribution as this Dorado catfish, okay? So it's living along rivers, okay? It's a very sort of linear type habitat. They can, they're sort of built to move. This is flooded forest part of the year, so they need to get out of there. So they're, just think, if I'm a bird, I live in flooded forest, I need to be able to move around more. So we predict anyway that there'd be better dispersers versus something like these toucans that are in upland forest and not associated with rivers. And so here's some actual predictions. You just expect that the birds in the upland forest, will just have more genetic differentiation, more genetic structuring than these floodplain Varzea forest birds, that need to move around a lot, okay? And so the experimental design here was to pick 20 pairs of species. And so again, these are going to be closely related pairs. Some of them are sister species. Some are congeneric. Morphologically, they're basically the same. Okay, so there's nothing like wing morphology at play, really. The main difference is really just that one occurs in upland forest, the other's in flooded forest. So it's really a habitat difference that we're looking at here. And so I'll show you a couple slides of results, but basically it's exactly what you'd expect. So the upland forest bird, and there's just an example with spot-backed antbird. I love antbirds, so they'll show up a lot in this spot. It's got more genetic diversity, however you want to measure it, okay? And then if you use a program like Structure, BAPS, to look at subdivision within a species, you'll see that the upland forest bird has sort of 3 defined populations, whereas this floodplain bird is just pan-mictic kind of thing. So another situation where what the bird does for a living, how it lives its life, its natural history, is having an impact on its population genetic structure. One reason that's important is because population genetic structure actually has long-term ramifications when you look at deep evolutionary time. There actually is a correlation between the two. I'll talk about that next. So, next project is that we amassed this massive, basically every mitochondrial data set on birds that had been published is in this data set. And so from it, we were able to get, it's got phylogeographic data with very tippy tips, okay, so sort of speciation rate. And then we also have the phylogeny, so you're able to get a speciation rate measure at deep time. The reason that's important is because if you look at the literature, there's expected to not be any sort of association between the speciation rate that you see at the tips versus at deep time, because at deep time you've got the extinction, right? But in between the tips and deep time, a lot of extinction has occurred. All right, so in terms of the latitudinal gradient, so this study encompassed all sort of New World birds we could find. You can see that there's a lot more genetic structuring within the tropics, okay? And so you can take that however you want. Maybe it has something to do with dispersal ability. We don't really know, but point is there is a gradient there in sort of the amount of within species variation that you see. The other analysis that we did was to essentially look and see, is there a correlation between the population genetic, so the speciation rate that you see at a really fine scale, versus the speciation rate that you would measure like deep in a phylogeny for that sort of same lineage. And it's a little bit hard to see from this, but basically there's a positive correlation, but it's only really significant in tropical birds, okay, and not in temperate birds. So in tropical birds, essentially there is a signature, a correlation, albeit weak, between the population genetic splitting rate, the rate that populations are formed, and the eventual speciation rate that you see in deep time. So what's going on at the population level can have deep ramifications. All right. Shift gears a little bit. So this is a project that we're just wrapping up on. It's a huge neotropical radiation called the suboxene passerine. This is a cherry-picked slide because most of them are completely drab brown or drab green. It's a group of birds that has gone nuts in the neotropics, although there are a few species in the old world. If any of you are birders here, you'll know them locally as the Empids. You can see the rich diversity of morphology there. It's very impressive. But if you go to South America, this is actually an important group. Suboxenes are like the comparable group to the ocenes that we have here. So we've got blue jays, cardinals, and mockingbirds. Ocenes, it's the suboxenes that dominate there. Like this Rufus ornero is the national bird of Argentina, for example. They're spectacular birds. They're in every terrestrial landscape, from coastal intertidal zone, the wettest rainforest, the driest deserts, you name it, they're there. I mean, it's just, when they got to South America, they sort of filled everything. Montane woodpecker, wood creeper, sort of like a woodpecker. It's Peruvian recurve bill. This thing is specialized on dead bamboo stems, so it'll use that bill to slice up the bamboo stem, find juicy katydids in there. Surfs and clodies. This is the only passerine that's found in the intertidal anywhere. It doesn't have a salt gland, so Carlos Martinez del Rio at Wyoming has been studying how it deals with salt in its diet. White-bellied synclodies, highly endangered, fernereid that's found above tree line. And so this is sort of the diversity of birds that you find in there. So we've really been trying to sample every single species, and even within species where there's species or subspecies that we think are probably species. It's the biggest data set I've ever had the pleasure of dealing with. And luckily, there have been people to help with the analyses. I mean, so just, you can do various levels of filtering, so you can do it unconservative, and you've got like a 1.5 million base pairs, or you can be aggressive and do 500,000. I mean, I'm like someone who graduated with a 500 base pair Site B data set. I mean, so this is mind-boggling to me still. All of you grad students, if you're not getting 10 billion base pairs in your PhD, Anyway, no pressure. I've always learned, don't make jokes to grad students. They take it seriously. I'm joking. All right, so this is, basically it does a pretty good job at reconstructing family relationships, which is a tribute to the taxonomists. You start getting into the genus level, species level, there's a lot of paraphyly. So right now with our tree, we're trying to just figure out what all needs new names and what do we need to synonymize and all this sort of stuff. And I love doing the basic taxonomic stuff. So, I mean, if you're a bird watcher, I don't know if any of you are, but like Acadian Flycatcher, it's the local Empidonax that'd be breeding around here. So Empidonax is a famous genus of flycatchers. Right? After we're done, Acadian will be the only Empidonax that's left. So it's not related to the other ones, so we get to give it a new name. So hoping that annoys the birders out there. We're going to give it a really obnoxious name that's hard to pronounce or something. All right. So another cool thing, and this is sort of preliminary analysis that you can do, is look for shifts in rates across this big tree. And so, one thing I was very interested in was the army following antbirds, of course. And so one of the cool things, so we were able to see that it evolved 3 times independently within the antbirds. They've been doing it a long time, even though they're specialized on this one ant species. So it sort of seems like a vulnerable existence, but it seems to be doing pretty well. But this specialization isn't like a key specialization. It has not resulted in any sort of increased diversity or anything like that, beyond the fact that we don't have this up here. So that's how this has contributed to richer tropical diversity. All right, so as you can see from the band plot there, this little black dot, there are a few little rate shifts there, thank God. And then there's this other thing called cranioluca, which isn't, I thought it had a black dot. I needed to put a black dot there. But anyway, cranioluca does have an elevated rate shift, and that just means it has more species than it should given the amount of time, right? And so the question is, why would this one little genus that, as you'll see, doesn't look like much, it's not impressive that much, what's going on there, okay? And so, all right, here's the genus cranioluca. There's 28 taxa within it. I mean, they're cool little fernariids found in a lot of different habitats. Little spine tails. And one clue to what might be going on with them is that there's this one species, Cranioluca anticiensis, that has the most impressive size variation of any bird in the world, as far as I know. I'll put that caveat in there. All right, so Cranioluca anticiensis undergoes a threefold change in size within a geographic space about the size of California. I mean, just imagine a human being doing that. Yeah, that'd be pretty impressive. And so one mechanism that we thought, might be going on in the situation, we thought this is a pretty good test case for ecological speciation. So it's basically the idea that reproductive isolation between populations can accrue as a result of natural selection, right, on those populations, by adaptive divergence. All right, so it's a bird that's found in the Andes, as I mentioned. It encompasses actually a pretty wide range of elevations, but anywhere that you encounter it, it's only got about a 500 to 1,000 meter wide elevational range. That sounds confusing. Okay, so if you're in the low Ecuador, which is the extreme northern limits of it, it's found at 1,000 meters, but you'll find it from, say, 750 to 1250 there. Okay, and as you go further south, down to Lima, you can find it at 4,500 meters, so a substantial increase in the elevational range. But then there, you'll find it from, let's say, 4,250 to 5,250, something like that. So it's got extreme sort of elevational zonation like a lot of Andean birds do. All right, so I mentioned that it has this threefold size variation. There it is. And so the birds up in the north at about 1,000 meter elevation are the smallest ones, and the ones in the south at the highest elevation are the largest ones. And as I mentioned, all of that can fit within a space about the size of California. All right, so here's some data. And this just shows mass against a latitudinal gradient. Luckily, the Andes are basically a straight north to south gradient, and so you can line it up like that. And so mass is changing, basically with elevation as context. So it seems like mass would be under natural selection. So it does seem like, at least an onset, that it's a decent candidate for something like ecological speciation. It fits Bergmann's rule where, colder temperatures at higher elevations, the birds are bigger. Okay, exactly what you'd expect. So it's got all the hallmarks of natural selection, even though we can't really do any experiments that would show that. Sort of less important, but I want to mention they change in plumage a bit along that same gradient. All right, so if you look at mitochondrial data in these, sort of the take home here is that they're all pretty closely related, right? So it's not a lot of divergence within it. So we think, put big error bars on this, but all of this happened relatively recently. All right, so you've got all of these qualities: recent origin, unsorted genetic variation, strong gradient, genetic or size variation that's consistent with Bergmann's rule. So it sort of seems like a perfect case for ecological speciation in many ways. All right, so there's sort of two things that need to be fulfilled for this to be ecological speciation. First is that there has to be natural selection causing it, and this is hard to do in a system that you can't control, right? We can't, we could try, it's not worth it. You could try to do this in captivity, but it wouldn't work. So anyway, so you have to sort of take on faith that body size would be under natural selection. We did actually find one method in the literature that tries to do a formal test of natural selection. It basically uses the amount of genetic variation that you see as sort of a null expectation of variance, and then you can just ask, is the amount of variance that you see in this phenotypic trait, does it exceed that? And if it does, you're like, well, then that could be natural selection. Sort of paraphrasing, but that's sort of the essence of it. And so the results of the test show that mass, significant natural selection, significant, let's say it's consistent with natural selection to be conservative, whereas plumage isn't. All right, so, I would say we've sort of fulfilled the first part there. And then the next part is like, does natural selection actually matter for speciation? Does this size variation have any relevance at all to speciation? Is it causing genetic differentiation? Right? If it's not, then it doesn't matter. All right, so there is genetic structure. This is from a RADseq data set that we collected. And you can see, it's a little bit hard to see, but it basically follows sort of the geography of the landscape. And when we plotted this out, we were like, oh, this is really cool. This has got to be ecological speciation, because body mass was like perfectly correlated with one of the axes of genetic variation. But it turns out that basically there's an autocorrelation situation going on here, because if you have allopatric populations, along this gradient, that co-varies with elevation, and that co-varies with genetic variation. So we haven't shown anything here except basically isolation by distance. So that was sort of a letdown, but that was what it was. All right, so as part of this analysis, we tried to exclude any loci that might be under natural selection. We basically wanted to come up with a set of loci, RADseq loci, that we could call neutral for our tests that we were, to measure things like isolation by distance. So we basically got rid of outlier loci. We don't know if these actually are under selection or not, but to be conservative, we did that. Okay. And so when you plot it out, and I'm not going to go into details about how we measured distance, but we did it multiple ways, and basically tried to come up with a distance measure between populations that matched what the birds, sort of the paths they would actually take. And you can see that there's a very strong measure, pattern of isolation by distance. All right, we also did some niche modeling to try to determine, sort of how, sort of the influence of the environment, sort of the habitat distribution on genetic variation. And then also we've got mass in here. So basically you can think of these two things as ecological speciation, and isolation by distance is the boring null hypothesis that would need to be rejected. And all of these things are in some way associated together, right? You can see that genetic variation is associated isolation by distance. It's correlated to mass. It's correlated to elevation, sort of the environment. So there's a lot of covariation going on. So this was a chapter of Glenn Seeholzer's dissertation, and he dug out this cool commonality analysis, which basically, sort of the idea of it is that if you have a bunch of different, sort of like a multiple regression, you've got a bunch of variables that are impacting something, and you can try to tease apart which of those variables is actually sort of most important. And every talk should have a undecipherable slide, so this is my undecipherable slide. But basically the take home here is that you can explain something like 97% of the genetic variation by isolation by distance, so the null hypothesis, right? And so what that means is that there is no ecological speciation, okay? You've got natural selection driving the size divergence, but the genetic variation that you see is just purely isolation by distance. There's nothing beyond that really that can be explained by, that we would attribute to speciation. All right, so then what is weird, sort of wrapping up here, what is weird about Cranioleuca? So Cranioleuca is that little genus that's got the super rapid rate of evolution, rapid rate of speciation, and it's another question I don't have the answer to, but I wanted to throw out this model. So typically we think of the allopatric speciation model. You could characterize it as isolation, then divergence, right? Stage 1, continuous population. Stage 2, isolated by a mountain or something. Stage 3, they become species. All right, so here's an alternative model that we call speciation by extinction. And the basic idea here is that taxonomists, in practice, look out at the diversity of organisms, whatever that they are. And this is true for birds as well. And we're looking for discontinuities, okay? Breaks in morphology, that sort of thing. And so, and when we see those breaks, a lot of times those get named as species or subspecies. And so speciation by extinction really isn't like a speciation mechanism like you're used to thinking about. But imagine that in stage 1 you had a bird like Cranioleuca antecedentsis that has this insane size variation. It goes 3 times the size variation. And then imagine just, I don't know, a virus or something takes out the middle population. It did it like 100 years ago. If we look at that now, we'd be like, those are 2 radically different populations, 3 times size different. Definitely species, okay? You've just had a speciation event, sort of, if you can think of it that way, just by sort of a local extinction. So we came up with this, and then you can't come up with original ideas in evolution anymore. So I dug in the literature, and actually Robb Brumfield had coined the speciation by extinction, I think for a beetle or something that they had studied on the East Coast, where the intervening population of a beetle that showed, I could be screwing up, it may have been a fly, I don't remember. But anyway, it showed variation, and I think like some development, human conversion of the landscape had removed sort of the center of the distribution, so that you now had these morphologically distinct populations that had once been just perfectly connected by the intermediates, hence speciation by extinction. Maybe that's going on. I think personally extinction is this a very important thing that sort of gets underappreciated in at least a lot of our work on birds. Things can change very rapidly in the bird distribution world. So just where I am in Louisiana, Inca doves have expanded their distribution very recently. So, and there's a lot of other examples like that. And then I'll leave you with this. This is a tar seep in northern Venezuela, okay, 26,000 years old. That's a rusty blackbird, okay? If any of you birders out here, that's an exclusively North American bird. But in this tar pit, 26,000 years ago, are fossils of both rusty blackbird and brewer's blackbird, another North American bird. That's not a long time ago, so things change a lot in the landscape. There's also a bunch of extinct passerines in there. So I think we just have to always be cautious when we're making our interpretations about speciational history. And I've got a ton of people to thank. Mike Harvey's dissertation, hopefully some of you got to meet Mike Harvey when he was a postdoc with Dan here, and Glenn Seeholzer's dissertation research figured prominently in this, so I want to give a special thanks to them, and I want to thank you all for listening, and that's Grooveville Donnie. I don't know if I have time for questions or.
You have definitely time for questions, but I do want to make one announcement that I forgot to mention earlier, which is that Rob is giving another talk tomorrow at 11 a.m. I can't remember the title of your talk, but at RMC, so that's just a reminder. So I guess I'm check the shuttle schedule, whatever, but anyway, there'll be a talk at RMC tomorrow morning. So
It's something like running a university-based natural history museum, backyard birds, backyard successes, and backyard challenges. Something like that. So, anyway, any questions? Yes.
So I find your story about the opposite ant colors really fascinating. I've always wondered, what do you guys split up the nature? I mean, there are so many species. Do they all seem to be the same?
No. I'm glad you asked that. So there's been this wasn't planted. I love these army ant pollinators. So Susan Wilson, I can't remember who the advisor was, maybe Betty Loisel, and Ed Willis before her did a lot of cool studies following these things around. And so what you see is that the big ones, the genus Phlegopsis, will take the front of the swarm, okay, which is the most productive part of the swarm. It's where the ants are hitting the fresh leaf litter. And then the intermediate sized birds, like the Gymnopithys and Regmatorrhina, are back from there. But then there's, I don't know if you remember that little white plumed ant bird that has the little white crown. They use a different strategy. They're small, but they dart into the front, the productive part, and like eat as much as they can before they're attacked by the Phlegopsis, the big one, and they scoot out of there. There have been studies of just the productivity, like sort of arthropods per minute of foraging, and it's when they're at a swarm, it's one of the most, I mean, they're eating constantly. It's an amazing resource, and so there is a lot of competition there. And I mean, and there's ant butterflies that are specialized on the scat of the birds too. So I mean, it's like if I were a grad student, especially with these GPS monitors now. Anyway. Definitely geographically isolated, no idea on the reproductively isolated. If I had to guess, they wouldn't be if they came into contact with each other. And so I think the problems with that model is that why don't, let's say, a virus knocks out the intervening ones, if the habitat's still good, then they just get repopulated by one or the other. So it would, I think for it to work, it has to be something sort of more permanent, like building some condos or, something like that. But if you built some condos, let's say you did that, I mean, you would then have this you'd have like size variation that is comparable to every other good species in that genus and most other. I don't think any avian taxonomist would even question it, and so I just wonder, how that, how frequently that sort of thing may actually exist out there. I think it's probably impossible to test it, but I think it's an interesting thought exercise, maybe. Yeah. Glad to hear you say that. All right, good. Yeah, we're trying to get some feedback because we're writing it up now. And yes. Are you talking about the antbirds or which? Oh, that's another great question. And that's one that, so the answer is that when they split, sort of all of the outcomes you can imagine happen, okay? And the place where we're studying this now, and I think I'll talk about this a little bit tomorrow, is in the headwaters of those rivers. So you know in the Amazon, like the Madeira and the Negro, where they get narrow, all of those birds that are completely differentiated, good species, down where the river's wide, but where the river's narrow, they come together. And in some cases, they form a hybrid zone. In some cases, they competitively exclude each other. In some cases, they're syntopic. They've completed the cycle, so that they are reproductively and ecologically isolated. And so we're trying to get, my last rejected grant is basically to study those contact zones at the headwaters of the Madeira, the Negro, and the Amazon. It's a beautiful natural experiment. It would answer your question. Yeah. Yes. So do you mean, oh yeah, yeah. Well, and those lineages tend to be more depauperate in terms so the ones that disperse well tend to have fewer species. Is that what you're saying? Or maybe you had it. Yeah, so the ones that are good dispersers will tend to have like less genetic variation or less, if you're looking at genus, fewer species within them because of what you're saying. They're just homogenizing. But I don't know if that's getting at what. I might have said it backwards. Sorry. Dan.
So thinking about your result with different levels of genetic structure within species in the tropics and temperate zone, so how much of that could be explained by like taxonomic artifacts, or how do you actually control for differential interest and density of ornithologists?
That's a good question.
It's kind of a conservative bias in some ways from the perspective of like latitudinal gradient, right? Because actually those should be elevated and treated. They actually, if the tropics are undersplit, then you have way more species birds in the tropics, right? Like, how do you control for?
Yeah. How do you control for? I mean, so we did it three different ways. So one way the first and second way is that we use two different taxonomies. So one was, I guess, like the AOS version of the taxonomy, and then the second was sort of expert opinion on what subspecies were actually species. And then the third way that we did it is using one of these species delimitation methods to basically just give it the data and let it decide what the species were, what the clusters were. It doesn't entirely get rid of any potential bias because even there, at some point you have to decide what the units are, and so I think we sort of did as well as one could. There still could be a taxonomic element to that. But it's definitely undersplit in the Neotropics. So, as you say, it probably makes it more conservative, but it'd still be nice to not have taxonomy as covariate in it. It's a good point. Any other questions? All right, well, I appreciate it. Thank you.
Thanks.